Hello, my name is Constance Jeffery, and I'm an associate professor at the University of Illinois at Chicago. I'd like to thank the organizers for giving me a chance to talk about some of our research today, and I'll be talking about the expanding world of metabolic enzymes moonlighting as RNA-binding proteins. RNA-binding proteins are crucial for all aspects of RNA function. They play roles in processing of the initial transcript, as well as stability of the RNA and its localization, translation, modification, and interactions with other molecules. The RNA and RNA binding protein complex can also affect the function of the protein, such as in catalysis, as well as its stability, localization, modification, and interactions with other molecules. In recent years, a number of labs have been using the CLIP method to identify proteins that bind to RNA. This method uses in vivo cross-linking to covalently attach RNA-binding proteins to the RNA they bind to. Then the cells are broken open and the RNA is isolated along with the bound proteins. After these complexes are broken up, the proteins that had been binding to the RNA can be identified by mass spectrometry. So variations on this method have been used to identify thousands of canonical RNA binding proteins, as well as many additional or non-canonical RNA binding proteins. One thing that was somewhat surprising is the number of enzymes from intermediary metabolism that moonlight as RNA binding proteins. This includes all of the enzymes from glycolysis and the citric acid cycle, as well as many other enzymes in carbohydrate, lipid, amino acid, and nucleotide metabolism. This ability to bind to RNA is conserved across species, so it's likely to be very important. The number of proteins that have a second function in binding to RNA suggests that there's a profound crosstalk and coordination between metabolism and gene expression. These proteins are actually great candidates for regulating gene expression because they are sensitive to the levels of metabolites such as their substrates and cofactors, and they're also sensitive to other cellular conditions. A classic example is aconitase. This is an enzyme in the citric acid cycle that uses an iron-sulfur cluster in its active site during catalysis. When cellular iron levels are low, the iron-sulfur cluster is lost, and the protein undergoes a dramatic conformational change to reveal an RNA binding site. The protein can bind to mRNAs, encoding proteins involved in iron uptake, utilization, and homeostasis. So many of these proteins in intermediary metabolism have a second function in binding to RNA, but a lot of these functions have not been previously predicted. We can identify the catalytic function through a number of methods that, for example, using sequence homology or looking at the three-dimensional fold or the constellation of amino acids in the active site. However, it's much harder to predict the RNA binding functions. These enzymes in intermediary metabolism lack the canonical RNA binding domain shape. There's also not like a standard pocket or groove, such as what we might see in an active site or a DNA binding protein. We also don't have any specific amino acid patterns that are characteristic of a moonlighting RNA binding protein. So my lab is very much interested in learning more about these RNA binding proteins, and we're working on identifying the locations of the binding sites on the protein, as well as, we hope, getting some more of the structures of the enzyme RNA complexes. And we think that by providing more examples of specific binding sites and complexes, will provide more of the data needed for developing methods for predicting the RNA binding function as well as the location of the RNA binding sites. And as part of this project, we'll also be studying the factors that affect the enzyme RNA interactions and the effects of the enzyme RNA interactions on the RNA as well as on the protein activity. In addition to our wet work projects, I wanted to just mention a little bit about some of the online research projects that we developed for undergraduate and high school projects during the COVID pandemic. During the spring of 2020, when labs shut down during the pandemic, a lot of students lost the opportunity to work on research projects, and along with that, all the mentoring and training they need to be able to continue in STEM careers. So during that first summer, I quickly came up with a project for some of our undergraduates where they could help out in updating our MoonProt database, and that grew to include 7 undergrads from our school, other places in the US, and also internationally. And we also included 4 high school students and their teachers. And that project actually worked out pretty well online and remotely. We got a publication for the updated database, as well as students were able to write papers to finish out their summer programs, complete honors thesis, and it also became part of one of the grad students' PhD thesis. In this past summer, we continued to do online remote projects, this time as an NSF-sponsored REU in macromolecular structure and function. So 2 students were in my lab with that project, and they helped look at some of these proteins that have a second function in binding to RNA. So to summarize, through the work of many labs, dozens of enzymes in intermediary metabolism have been found to bind to RNA. And this is apparently part of an extensive network of connections between these enzymes and gene expression. But it's difficult to predict that these proteins have this RNA binding function, and it's also difficult to predict the locations of the RNA binding sites on these non-canonical RNA binding proteins. So we need a lot more information about the structures of the proteins with the RNA bound and the locations of the binding sites. And I'd just like to thank a large number of people who have been helping through the years with our Moonlighting Proteins projects and the MoonProt database, as well as the more recent projects involving enzymes that moonlight as RNA binding proteins, and especially the work of Nicole Curtis, Krupa Patel, and Pritha Das. And I'd like to thank you all for your attention, and I'd be happy to hear your comments and suggestions. Thank you.